In this lesson, we'll be comparing codominance and incomplete dominance. We'll be looking at the ABO blood group, the Punnett square, and other genetic diagram. We'll also be able to tell who is a parent of a particular child based on blood group. At the end of the lesson, students will be able to answer any question that is set on the topic in their external exam. Let's ride along. Incomplete dominance. In incomplete dominance, what happens is that a new phenotype is formed in the F1 generation. So this is a type of inheritance in which both alleles are expressed in the heterozygous form of the offspring to make a new phenotype. The new phenotype does not show the exact phenotype of the parent. So here we are crossing red and white impatient plant, and the new phenotype here is of course pink, and you'll observe this is in its heterozygous state. Let's look at the genetic diagram, and we're going through this again so you're able to recall. So the phenotype for the parent here is red and white with a genotype as is shown on screen. Never forget that you should always put in the gamete. There we have the gametes going in, and then we move on to random fertilization, or some might say segregation. So we're doing the exact cross for that which you saw earlier, and we're now mapping for the F1 generation. If you follow closely on screen, you're able to do likewise. There we have it. For the first possible offspring, they are both heterozygous, so they'll be pink. Heterozygous again, so offspring there will be pink. The phenotype will be pink. Pink plant, pink plant, pink plant, pink plant. Alright, so there we have all will be heterozygous with the genotype RW and phenotype being pink. Let's look at codominance. Now in codominance, the same is not true. While we're using the same impatient plant, what we have happening is that both parent phenotype is expressed in the offspring. This is a type of inheritance in which both alleles are expressed in the heterozygous form of the offspring. So there we're crossing the impatient plant just like we did in incomplete dominance, but in codominance, if you observe here, we're not having a pink plant, we're having instead the phenotype of both parent in the offspring. So we're seeing red from the red parent and of course white from the white parent. Let us look a little closer at this. So the comparison here, we're looking at codominance to the left of the screen and incomplete dominance to the right of the screen. Look at what happens. The phenotype of both parents is visible in the phenotype of the F1 generation in its heterozygous state. Always remember, in the heterozygous state. Now when we look at the incomplete dominance, a new phenotype is formed. So you're realizing here that we have a pink plant instead of a red and white plant, and they are both in the heterozygous form. So this is a nice comparison of codominance and of course incomplete dominance in impatient plants. Let's go a little further in the lesson. So let's look at the cross. The cross is pretty similar. And we're looking at the genetic diagram here. We're going to be looking at the Punnett square as soon as we get to ABO blood group. So please be reminded that your genetic diagram should be laid out exactly like it is laid out here. And if we are not using this genetic cross and we're going to be using the Punnett square, at least we must take it to the point of gamete, and the Punnett square will of course reflect random fertilization, the F1 generation, and of course the genotype of the F1 generation. So as on screen, that is what is expected. Here we are having plants with red and white in their heterozygous form, and of course this is for codominance. Had it been incomplete dominance, then a new phenotype, so the offspring would have been pink, but here it is of course red and white. The ABO blood group as we treat with codominance. Now the ABO blood group, here we have three different types of alleles. We have allele A, B, and O. And of course, I there represent the loci where it is found. A is dominant to O. B is dominant to O. However, AB is codominant. While there is 3 allele, only 2 can be present at any time in one cell. So while we have 3 alleles here, only 2 can be present at any particular time in the same cell. That is something that we need to take in consideration when we are doing the genetic diagram to determine the F1 generation. So these are the possible phenotype and genotype for the blood group AAs. The phenotype for AA is going to be blood group A, AO, A, BB, phenotype B, BO, phenotype B. And you're realizing there when we have A and O or B and O, the B or the A is the phenotype because they are dominant to O. Where there is AB, then we understand that is codominant, so both will be expressed. Here we are having O, and of course this is the recessive allele, unlike the other crosses that we have done before. So we're putting this in another genetic diagram. There we have the gametes. Now we have random fertilization. Pay close attention to the cross. Shouldn't be getting this one incorrect. If you observe, we're using the color code for where we're getting the alleles from, or the gamete in this case. Look at what is happening. We use a color code. Now really, what we use a color code, but usually the dominant allele is written in front of the recessive. Here the recessive being O, the dominant being B. However, for simplicity, we showed it to you this way. Alright, so the phenotype here is going to be B, 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 because of course B is dominant to O. Let's look at the Punnett square. So there we have the gametes. Here we're using the Punnett square to do random fertilization. Please look at the construct. Then we have here color coded where we got those gametes from. And here color coded again. Here we do our cross. Blood group there being A. Blood group there being A again, being that A is dominant to O. Blood group here being AB, being that they are heterozygous. And blood group here being B, being that B is dominant to O. The F1 generation phenotype is going to be 2A to 1B to 1AB, and that's going to be the phenotypic ratio. However, if we look at the genotypic ratio, it's going to be a little different. It's going to be 1AA to 1AO to 1BA to 1BO. And of course here we are stating what the likely genotype would be for the F1 generation. Just a slight difference there. Still using the same letters, however the interpretation is a little different. I do hope that you enjoyed that presentation and that we want you to continue watching our program CSEC Biology, the cover page, or one might say CSEC Biology TCP with Mr. Wilson. We want you to like, share, subscribe, and of course leave a comment. Is there something we could have done differently, or tell us, did you enjoy this presentation? We want to also assist you in finding our product. You're going to click on that blue icon that says CSEC Biology TCP, which is usually left of the name for your video, and that will take you to the back end of the channel to visit our many videos. There's also this white icon that gives the suggested video. You could click on this as well, and it will give you the video that comes up next. Also, if you want to subscribe, you can click this icon here to the bottom of the screen. It says CSEC Biology TCP. It's yellow, and you can subscribe there or hit that red subscription button. And remember, when you do, to hit the notification bell so you'll be informed as soon as there's a new publication. You'll also see some videos on the screen, 4 possible videos from my collection. Choose one that will help you. Thanks, and continue watching.